The food that goes in must come out. But what happens in the stages between in the digestive system is the mystery we're going to solve. This lesson is all about the digestive system and how it functions to break down the food once we consume it. So our digestive system has four main functions. The first function is ingestion, so obtaining and eating the food. Digestion, breaking the food down into molecules small enough for the body to absorb. Absorption, so cells lining the digestive tract absorb nutrient molecules and then they travel into the bloodstream. And elimination, so where our undigested food material is released into the environment. So our digestive system is kind of like a hollow tube that starts with the mouth and ends with the anus. So we ingest our food through our mouth, it travels through our esophagus into our stomach, from there into the small intestine, large intestine, the rectum, and then eliminated through the anus. And it measures between 6.5 and 9 meters in length. Lots of this is in the intestinal system. Together, this muscular tube is known as our gastrointestinal tract or GI tract. Begins with the mouth and travels through the body, ends with the anus. Once we pass the mouth, we have the pharynx and the esophagus, the stomach, small intestine, large intestine, rectum, and anus. There are several major organs along the digestive tract that aid in digestion but are not part of the GI tract. So they're connected, but food does not travel through them. These are the salivary glands in our mouth, the pancreas, the gallbladder, and the liver. So food does not pass through these organs whatsoever. The digestive process involves mechanical digestion, so physically breaking down food, chemical digestion, using chemicals to break down food, and absorbing nutrient particles into our bloodstream. So the first task of the digestive system is to break down food into a fine pulp. So this begins in our mouth. Doing this increases the surface area of the food and exposes more of it to the digestive chemicals in our digestive system. The process of mechanical digestion breaks food into tiny pieces without changing the chemical structure of the food. So it changes the physical structure, but chemically it's still the same as it was beforehand. The second task of the digestive system is to chemically break down food into smaller and smaller particles. So molecules must be small enough and chemically simple enough to be absorbed into the bloodstream. So large macromolecules are broken down into smaller nutrients. So our starches are broken down into monosaccharides, or those single sugar particles, proteins into amino acids, and lipids into the fatty acids and glycerol. The last task of the digestive system is to absorb the small molecules and pass them into the bloodstream so they can be distributed throughout the body. The circulatory system is going to take nutrients from the digestive system and deliver them to all the cells in our body. So the digestive system starts with our mouth, and this is both where mechanical and chemical digestion begin. So chewing is the first step in mechanical digestion, and then salivary glands produce saliva, which is a mixture of water, mucus, and the digestive enzyme called salivary amylase, which is where chemical digestion will begin, which mixes with the chewed food. So the saliva is produced by a set of glands located near the mouth. So the tongue helps to keep the food between the chewing surfaces of the mouth. So we use our tongue to move our food between our teeth, between your gums if you're have had all your teeth pulled, or if you're a baby, and against the hard palate, so the membrane, the bony membrane covered roof of the mouth. So we use our tongue to move food all around. The mucus in the saliva softens and lubricates the food and helps hold the food kind of together in a ball known as a bolus, which is then going to be easy to swallow. In addition to the salivary glands, we also have taste buds located on our tongue, which are chemoreceptors and specialized cells which detect certain tastes when those food chemicals are dissolved into saliva. So we can taste salty, sweet, bitter, and sour very easily. So in the saliva, we also have enzymes that will begin to kill the bacteria and begin the process of chemical digestion by breaking down starches into sugars. So these are going to be kind of our simple starches be broken down into simple sugars. The enzyme salivary amylase begins chemical digestion by breaking them starches into disaccharide maltose. And then once the teeth and salivary glands have completed their initial mechanical and chemical digestion, our food ball bolus is ready to be swallowed. So once it's swallowed, it enters the esophagus. So this moves past the pharynx into the stomach by way of the esophagus, which is a roughly 25 centimeter long muscular tube. So we have the bolus of food, it goes into the esophagus, and then muscles will slowly constrict in the esophagus, moving it down into the stomach. So in the pharynx, the gastrointestinal tract and the respiratory system cross each other. So as the tongue moves food into the pharynx, it presses down on a small fat flap of cartilage known as the epiglottis. So the epiglottis is going to cover our trachea to allow food to go over that and down into the esophagus. So it closes the entrance to the respiratory tract and guides food into the esophagus so that we're not inhaling food. Now, occasionally people do choke, whether or not they were surprised, or they fell, or they inhaled when they had food in their mouth, or whichever reason. And this is the process that kind of doesn't work properly in that point, is the pharynx. In the pharynx, that epiglottis doesn't completely cover the esophagus, or the trachea as we're swallowing food. So once the bolus enters the esophagus, muscles in the esophagus wall move food towards the stomach. There's two muscle layers, one that runs the length of the esophagus and one that runs around the tube. So these waves of muscular contractions called peristalsis move food down the esophagus. So our muscle contractions want to move in order to move food down. Imagine trying to get the last toothpaste out of a toothpaste tube, right? We want to start at the bottom and work our way to the top. We can't just randomly squeeze the tube and hope that it's going to come out. Now, at the bottom of the esophagus, there is a cardiac sphincter, which allows food to pass into the stomach, but ideally, we don't let food move from the stomach back up into the esophagus. So you can see there where we have relaxed muscles, contract muscles that are going to constrict above the bolus of food so that it keeps pushing it down, moving into our stomach. And then we have that sphincter at the bottom of the esophagus or top of the stomach, gastroesophageal sphincter. It's also been called the cardiac sphincter. And if this sphincter is loose or functioning improperly, or your stomach's too full and food does pass back up into the esophagus, this is when people experience heartburn. So the stomach is a muscular sac with a thick expandable wall. So our stomach can expand and contract, located in the upper left side of the abdominal cavity below the diaphragm. So it's actually quite high up in our abdominal cavity. And it can expand to contain about 1.5 liters of food. And in the stomach, food continues to undergo both physical and chemical digestion. So that mechanical, physical digestion will occur when the stomach walls are going to contract strongly, mixing and churning the food. So kind of imagine like if your stomach was a washing machine and all that food is in there swishing around and your stomach is churning it with its muscles. And these contractions are responsible for the kind of growling noises that our stomach makes, which are incidentally the loudest when we have an empty stomach because there's nothing to absorb those sounds. Now, when it comes to chemical digestion, the stomach is lined with millions of cells that secrete a variety of fluids. Some of them are gastric juices, which is hydrochloric acid, mucus, pepsinogen, which helps digest proteins. And the inner lining of the stomach is thick, mucousy, and contains epithelial cells. And so these cells are protected by a mucus lining in the stomach so that the stomach doesn't digest itself because of that low pH hydrochloric acid content. And the membrane of the stomach has a bunch of small openings called gastric pits, which contain gastric glands, which is what's going to release that gastric juice, that hydrochloric acid, pepsinogen, mucus into the stomach. And they each have their own specific juices that they release, but that mixture is going to be that gastric juice altogether. So that digestive fluid contains hydrochloric acid, which lowers the pH of the stomach to between 2 and 3, which is extremely acidic. It helps, again, to kill pathogens. It converts pepsinogen into its active form pepsin, which is going to break down those protein molecules into smaller amino acids called polypeptides. And again, when we remember talking about enzymes, that the enzymes are often pH specific. So if we don't have that hydrochloric acid in our stomach, those stomach enzymes aren't going to work properly. So all of our cells are made of protein. So I guess the enzyme pepsin could potentially break down the cells of the gastric glands, making the stomach digest itself. But the cells of the stomach protect themselves by producing an enzyme in its inactive form called pepsinogen. So when we have that gastric juice first pumped in, the enzyme pepsinogen is inactive. Once it's out of the cell membrane and past the mucous lining of the stomach and it comes in contact with the hydrochloric acid, it's now active. So when the enzyme is produced in the gastric glands, it's inactive until it makes its way into the juices of the stomach where the pH is lower, is when that pepsinogen becomes the active form of pepsin. So mucus, part of the gastric juices, lubricates the food so it can again easily travel through the digestive tract. It also coats the walls of the stomach, protecting them from the acid and the other digestive juices. The stomach wall cell lives are very short and are replaced about every 3 days because they're in a highly acidic environment with, again, those digestive juices. So it's a very rapid cellular turnover. So after food's been in your stomach for about 3 to 4 hours, it's reduced to a soft pulp called chyme. So this is a thick liquid made up of partially digested proteins, starches, vitamins, all the things that we had eaten, partially digested, mixed in with those gastric juices. At this point, the sphincter at the bottom of the stomach, known as the pyloric sphincter, between the stomach and the small intestine, opens and allows small amounts of chyme to pass into the small intestine. By the time this happens, most of the proteins have been broken down into polypeptides. Sugars and fats have not yet been chemically altered besides from the salivary amylase in the stomach. So in the stomach, the sugars and fats are not chemically digested, only mechanically digested. So once in the small intestine, the majority of the chemical digestion is going to take place. The small intestine is about 7 meters long, but its diameter is only 2.5 centimeters. So it's smaller than the large intestine, and it consists of 3 separate components. The duodenum is the first section, and it's about 25 centimeters long. The jejunum is the middle section, about 2.5 meters long, and the ileum makes up the remainder. So as chyme is pushed through the pyloric valve into the small intestine, it enters the duodenum, which is, again, that top 25 centimeters of the small intestine. This is where most of the chemical digestion takes place. So in the duodenum, the carbohydrates and proteins are continued to be chemically digested, and the breakdown of fats begins. So though the stomach might absorb some water, some specific vitamins, medications, and alcohol, most of the absorption from digestive materials occurs in the small intestine. So the small intestine's primary role is chemical digestion and absorption. In the small intestine, there's a lot of finger-like projections called villi, which line the walls of the small intestine and increase the surface area by about 10 times. So all these small projections, and then that chyme and that fluid will go between all of them, increasing the surface area for absorption into our circulatory system. Each of the villi also has smaller projections called microvilli, which further increase the surface area. And each of these villi have a capillary network of blood vessels, small lymph vessels, where monosaccharides and amino acids are absorbed and fats are absorbed into our circulatory system. And this can occur by passive transport, so requiring no energy, or active transport. So in the small intestine, this is where now those auxiliary organs, the liver, gallbladder, pancreas, also come into play in our digestive system. So the role of the pancreas is twofold. One of them is to, it adds sodium bicarbonate to the hydrochloric acid into the chyme to neutralize the pH of the chyme as it moves from the stomach into the small intestine. And it also secretes a chemical called secretin, which helps absorb, when the pH of the small intestine is lowered by the acid, a chemical called prosecretin is turned into secretin, which is carried to the pancreas then. When the pancreas gets this chemical messenger, it releases bicarbonate ions, and these raise the pH from about 2 to 9. So now we have a slightly basic environment, which then deactivates the pepsin, turning it back into pepsinogen. So we're not going to have digestion of our small intestines by our stomach acids. Secretions of the pancreas also contain enzymes, which are going to help break down the proteins, carbohydrates, and lipids. Two other auxiliary organs are the liver and the gallbladder. So the liver has four main functions: the synthesis, breakdown and conversion, storage, and detoxification. The liver is always producing a fluid called bile. Bile is a greenish liquid that contains bile salts and pigments from old red blood cells. So it's kind of like a greenish yellow color. And bile is important because it continues the mechanical digestion by emulsifying fats. So bile allows our fat molecules to become kind of emulsifies them, like soluble in water. So if we had a bunch of oil floating on water, we added bile, it would help those oil particles break down and become more soluble in water, also known as emulsification. So this increases the surface area of the fats so that the lipases can work more effectively. So this doesn't change the structure of the fats. So it's considered physical digestion or mechanical, not chemical. So it breaks down the globule of fat into smaller parts, so it's more has more surface area, but it doesn't change the fat component itself. It is continually produced by the liver, but it's stored in the gallbladder. And when fats are detected in the small intestine, a hormone called cholecystokinin is going to be released into the bloodstream. Now, this hormone is going to be carried to the gallbladder, which is going to trigger the gallbladder to release bile into the small intestine. This is going to help break down those fat molecules into smaller molecules with more surface area. The liver also stores glycogen, which is an energy reserve, vitamins A, B12, and D. The liver can also detoxify harmful substances by turning them into less harmful products. And many harmful materials dissolved in the blood, such as alcohol, pesticides, carcinogens, are detoxified by the liver. The liver is also a regenerating organ where if you have liver damage, change your diet, change your lifestyle, make some changes, can become more healthy than it was previously. Or if you gave somebody part of your liver as a living transplant donor, your liver will grow to become closer, if not the same size it was before. So very cool. Now, the large intestine is going to be one of the last parts of the digestive system where our GI tract. And by the time food reaches the large intestine, our chemical digestion is complete. Absorption is complete in the small intestine, and any undigested material then is going to leave the small intestine through another valve and enter the large intestine, which is about 5 meters long and 6 centimeters wide. So twice as large in diameter as the small intestine. The primary job of the large intestine is to absorb water and water-soluble vitamins and excess salts from any remaining digested material after digestion has occurred. So when most of the water has been removed from the undigested material, now we have feces left over. And we have that systemic contractions again called peristalsis, where we're going to propel the feces through the large intestine towards the rectum, which is the last few inches of the large intestine, before they're eliminated out through the anus. So we also have an organ called the appendix located near the junction of the small and large intestine. It's a finger-shaped pouch which doesn't serve any known function. If it becomes infected with bacteria resulting in appendicitis, it must be removed. In addition, your colon is going to be filled with billions of bacteria such as E. coli, which aid in absorption and produce vitamins B12 and K, as well as some amino acids. There can be more bacterial cells in our intestines, individual cells of bacteria in our intestines than our human cells in our whole body. So it's really quite amazing how our digestive system works together. Perfect. Well, I hope that you enjoyed the lesson on the structures of the digestive system, and I'll see you guys next time. Bye.